Hello.
Hello.
Hello. Vimpistundandi?
Good, sir.
Yeah.
Good. Sir, very good morning, sir, and good morning, all of you. Wish you happy Ugadi.
Thank you, same to you.
Myself, Dr. Kishore, Assistant Professor, Department of Zoology, Dr. B.R. Ambedkar University. A very hearty welcome to this webinar, which is organized by Department of Zoology and respected VC sir, Registrar, Health Director, Dean Faculty of Science, and today's special guest, Vijay Kumar Garu, and all directors, deans, faculty members, students, and participants. A warm welcome to all for this program. So already we are conducting a webinar series. This is the third webinar. Sir Vijay Kumar sir already, Vijay sir already delivered a lecture, first lecture, and this is the second lecture from sir. And I thank each and everyone for attending this program. And I hope this webinar would be helpful to all. I will now hand over the session to Vijay sir to deliver the lecture. Sir, please continue.
Thank you. So today I'm talking about protein sorting, or it's also called as protein targeting. You know, proteins are very important biomolecules and required in our body. So major biomolecules in our body are carbohydrates, fats, and proteins. Carbohydrates and fats, they provide only energy. Whereas the proteins, they perform different functions in our body. For example, you see here, they function as catalysts, enzymes. Yeah. So in our body, different chemical reactions that take place, and all the chemical reactions are normally catalyzed by enzymes. For example, here you can see the lactate dehydrogenase, or say simply for digestion, in the mouth, we have saliva. In the saliva, you have an enzyme called amylase. So in this way, without enzymes, our body cannot function. So this is the main function of proteins. And there are proteins perform different functions. For example, non-catalytic. These are catalytic proteins. They catalyze enzyme reactions. And there are non-catalytic proteins like, say, carrier proteins. Carrier proteins, they transport molecules from, say, blood into the cells, or cells into the blood, vice versa. And there are transport molecules like, say, hemoglobin, myoglobin, which transports oxygen. Then you have receptor proteins. So these receptor proteins, they act as receptors for different molecules. Normally, all hormones, they bind to receptors. So these receptors are very important. And then you have membrane transport. For example, in the enzymes, see in the membrane also you have enzymes like sodium, potassium, ATPase. And you have structural proteins, which give strength to the body. For example, in our bones, you have collagen, which is a strong protein. Similarly, for movement, you have proteins which are called as contractile proteins. Say in the muscles, you have actin and myosin. Even in flagella, you have a protein called as tubulin. And proteins, they also act as hormones. Say like, for example, insulin, glucagon, adrenocorticotropic hormone, all these. Now, in addition to that, they also provide immunity to us in the form of antibodies. So in this way, proteins perform various functions, unlike carbohydrates and fats, which provide only energy. Now, see how these proteins are maintained. See, normally we eat proteins in our diet. So these proteins are hydrolyzed, that is digested in the stomach and intestine, and they liberate amino acids. Proteins cannot be directly absorbed because they are macromolecules. So these amino acid, this is called as amino acid pool. This amino acid pool is used for protein synthesis because there will be turnover of proteins. No protein will be there permanently in our body. So every protein has a half-life, certain amount of time. After that time, it is hydrolyzed and again resynthesized. That is called as the turnover of proteins. So if the amino acids are in excess, then they're catabolized and used for energy, and urea and carbon dioxide liberated. Okay, so this amino acid pool helps in synthesizing new proteins in the body. So whatever proteins we eat, they are digested and amino acids are liberated. These amino acids transport in the blood to different cells, and according to the requirement of the cell, the proteins are synthesized. Now, a typical cell, a typical mammalian cell contains 10,000 different types of proteins, whereas an yeast cell, it will be having nearly 5,000 proteins. And these proteins, majority of the proteins, they are synthesized on ribosomes inside the cell. In the blood, they cannot be synthesized. Only inside the cell, you have machinery for protein synthesis, that is ribosomes, and then you require different mRNA and pool of amino acids for the synthesis. So they are synthesized in ribosomes. And these ribosomes are again, they may be present in cytoplasm or they may be attached to the endoplasmic reticulum. So if they are free, they're called as cytosolic ribosomes. If they are attached to the endoplasmic reticulum, they are called as the rough that gives you the rough appearance. That's why it's called as the rough endoplasmic reticulum. Now, all these proteins, they are synthesized in the cytoplasm of the cell, but they are delivered to different compartments. See, in our cells, you have compartments like, say, mitochondria. Mitochondria is a compartment for oxidation of foodstuffs, particularly carbohydrates, which produce acetyl coenzyme A. This acetyl coenzyme is oxidized to produce ATP. So that is compartment for oxidation. Similarly, you have a compartment, for example, lysosomes. They'll be having hydrolytic enzymes. So the function of these lysosomes is to hydrolyze different proteins. So in this way, every compartment has, see, this is called a compartment. In prokaryotes, for example, in bacteria, you don't have any compartmentation. Only in eukaryotes, you have compartmentation. So these proteins which are synthesized in the cell, in the cytoplasm, they are delivered to different cell organelles, or they may remain in cytoplasm, in the aqueous compartment. Or these proteins may also be secreted outside the cell as secretory proteins. For example, say yesterday we have seen about the insulin. Insulin is secreted from the beta cell into the bloodstream. Or digestive enzymes like pepsin, trypsin, chymotrypsin, they are synthesized inside the pancreatic cells, acinar cells, and then they are released into the digestive tract. So in this way, there will be secretory proteins. So whether a protein to remain in cytoplasm, or it should remain in it should go into the membrane, or it should go to the mitochondria, or it should go to lysosomes. So in this way, it is decided by the protein itself. This sending of the protein to different compartments is called as protein sorting or protein targeting, because the protein should go to a particular cell organelle. That is called as protein targeting. So for example, here the hormone receptors, proteins, and the transporter proteins. Transporter proteins are very important because last class I told you about the glucose transporters. Glucose is a water-soluble molecule. It cannot pass the membrane because the membrane is made up of lipids, hydrophobic, whereas the glucose is hydrophilic. So for that, it requires a transporter that is called as glucose transporter. There are nearly 12 types of glucose transporters, depending on the type of the tissue. So you have receptors, hormone receptors. They are also present on the cell surface. So that these receptors are synthesized inside the cell, but they are targeted onto the surface of the cell. So this is called as protein targeting. Now, for example, some enzymes like RNA and DNA polymerases. See, RNA and DNA polymerases, they are situated in the nucleus. But these enzymes are synthesized in cytoplasm. From cytoplasm, they should be transported into the nucleus. That is called the import of these enzymes into the nucleus from the cytoplasm. Now, in addition to inside cells, you have extracellular matrix. That is the material which is present between the cells. So the material is more in the case of connective tissue, whereas it is less in the case of epithelial tissue. In epithelial tissue, you have just a basement membrane, which is connective tissue. And between two cells, you have cementing material. Whereas in bone, you have more matrix and less number of cells. So, but these materials like collagen and other things, they should be secreted from the cells into the matrix. So they are extracellular, so that should be released outside the cell. So the delivery of newly synthesized proteins to their proper cellular destinations, it is called as protein targeting, or you can say sorting. Sorting means just like you have to separate the different types of proteins, which they should go into different compartments like mitochondria, lysosomes, Golgi, or say other macrosomes like that. So this is the topic which I'm going to talk about, and how the proteins are targeted or into the different compartments. Now, if you see this diagram, the first one is cell-free protein synthesis. Cell-free protein synthesis means cells will not be there. That is, if you take a tissue, then homogenize it, that is grind it, then the cell membranes are ruptured and cell organelles are liberated. So in that, you'll be having all components except cells. So that system is called as cell-free systems. So in the cell-free system, so if you allow an mRNA to synthesize the proteins, so here the protein that is synthesized will be having some extra length. Here you can see in the red, the normal proteins, they will not be having this extra length. But if they are synthesized in cell-free systems, you have extra lengths. So complete proteins, they have an extra length of amino acids. So what are proteins? Proteins are nothing but they are the chains of amino acids having two ends. One end is called as amino end, the other one is carboxyl end. There will be two ends. So these two ends, but one end will be having extra length of protein when compared to the normal protein. Why this? So if you synthesize the same protein in cell-free protein with the microsomes. What are microsomes? Microsomes are, suppose if you homogenize the cell, you have in the cell, you have endoplasmic reticulum, which is very long, that breaks into small vesicles and forms small round vesicles, which are called as microsomes. If the microsomes are there, then the protein will be of correct length, not extra length. In the upper diagram, you will have some, see this red extra length of protein, whereas here you don't have any extra length. Mature protein without any extra length. This extra length is called as signal sequence. So this is called as co-translational transport. That means translation means synthesizing a protein from mRNA by ribosomes is called as translation. And co-translational transport, at the same time, the proteins are transported into the microsomes. That is called as co-translational transport. Now, what is signal sequence? See, just now we have seen that there will be an extra length of protein that is called as a signal. So this signal is important for the import of proteins. So in an experiment, an mRNA encoding a secreted protein. Secreted means that is delivered outside. Protein was translated by ribosomes in vitro. In vitro means outside the body. In vivo means inside the body. So you can take the cell-free system, introduce mRNA for a protein, and in vitro with microsomes. When microsomes were omitted, when the microsomes were not there from the cell-free system, the protein synthesized was slightly larger, longer, you can say, longer than the normal secreted protein. And this extra length being the N-terminal leader peptide. See, I have told you proteins will be having two lengths, two ends, you can say. N-terminal end, there is N-end or amino end, C-end or carboxyl end. At the N-terminal, you'll be having some extra length. This is called as the signal sequence, which signals the direction, it directs the protein to enter into the endoplasmic reticulum. So in the presence of microsomes derived from the rough ER, however, a protein of the correct size is produced. So these results were explained by signal hypothesis. Signal hypothesis means the protein that is synthesized without microsomes having an extra length, whereas the protein with the microsomes having the correct length. So the extra length is the signal sequence or reader sequence. And this hypothesis was called as signal peptide hypothesis. And this extra length is cut off in the vesicles of the microsomes by an enzyme that is called as signal peptidase. So the signal sequences, they were first described or discovered in the early 1970s in secretory proteins that are translocated across the ER membrane as the first steps toward their eventual discharge from the cell. That is before secreting outside the cell. So they should enter into the endoplasmic reticulum, and then there will be a pathway, and this pathway I will explain you. So here you can see, this is the diagram. These are the ribosomes. Normally ribosomes, when they are not synthesizing any protein, the small subunit and large subunit, they will be separate. They will not be in the combined form. They will be free. These are the free ribosomal subunits when they are not synthesizing any protein. So when they are synthesizing a protein, that means they will be having mRNA. Then the small subunit and the large subunits, they bind and the proteins start synthesizing. And here you can see a green portion and a red portion. This red portion is called a signal sequence. This signal sequence directs the ribosome to bind to the endoplasmic reticulum. If the signal sequence is not there to the protein, the ribosome will be free in the cytoplasm as a polysome, but it will not attach to the ribosome, this endoplasmic reticulum. So this red is called a signal sequence. When the protein is synthesized, the length increases. Here you can see here, this red portion is cut off, that is cleaved by an enzyme that is called as signal peptidase, cleavage of signal peptide by the enzyme signal peptidase, and then the protein is liberated or discharged into the cavity or lumen of the endoplasmic reticulum. And then again, the ribosome becomes free and it circulates. So this hypothesis is called as signal peptide hypothesis. It was proposed by Blobel and Doberstein in 1975. So when the ER signal sequence emerges from the ribosomes, when the ribosome is synthesizing the protein, first you will get from the protein, the signal sequence comes, and this signal sequence directs the ribosome to a translocator. This blue one here is a translocator, which helps in the translocation of the protein from ribosome into the endoplasmic reticulum. So this translocator on the ER membrane that forms a pore, a hole is formed by this translocator. Through this hole, the protein enters into the cavity of the endoplasmic reticulum, and then the signal is cut off and the protein is liberated. So the signal sequence is clipped off during this translocation by a signal peptidase, and the mature protein is released into the lumen of the endoplasmic reticulum. So we now know this hypothesis is correct in outline, but this has been, now you see here, these are the ribosomes, which are free in the cytoplasm. Now this red one is mRNA. And the group of ribosomes, they're called as polysomes. Now they are synthesizing a protein. This blue colored one is a protein. So if this protein has a sequence that is called as the ER signal sequence, this red one here, then the ribosome is directed to bind to the endoplasmic reticulum. Now, after the synthesis, see here, the red portion is cut off. That is signal peptidase enzyme cuts off the signal sequence and the protein is released. And these proteins in the endoplasmic reticulum, either they may enter the membrane, or they may enter the Golgi complex, or they may enter the plasma membrane, or they may be transported to lysosomes. So any protein that is synthesized on the rough endoplasmic reticulum is transported to Golgi, membrane, lysosomes. This is the compartmentation here. Now, here you don't have any signal sequence. When the signal sequence is not there, then after the synthesis, the protein remains in the cytoplasm itself. It will not go anywhere. There is a location cytosolic protein. Or it may enter mitochondria, or peroxisomes, or the nucleus, or in the plants into the chloroplast. So in this way, it is decided whether the protein should go into these areas or into these areas by the signal sequence endoplasmic reticulum, which is having here ER signal sequence. So this is the mechanism here. You see, all normally it is called as a common pool of ribosomes, which are free, they are not bound. And when they come in contact with the mRNA, they form a group of ribosomes, which are called as a polyribosome. This polyribosome, if it contains the signal sequence, it attaches to the endoplasmic reticulum. And this is the mRNA, this is the protein, this is the ER signal sequence. So then the protein is synthesized and released into the endoplasmic reticulum. Now these are the proteins, how they go. Free ribosomes in cytosol, that means which are not having any ER signal, that is endoplasmic reticulum signal, they are synthesized, the ribosomes become free and they synthesize the protein, which either goes into nucleus, mitochondria, chloroplast, or peroxisomes. And if membrane-bound ribosomes are there, then this is the endoplasmic reticulum. Here, this is the translocator. Now, this is the protein that is being synthesized. This red one is the signal sequence, and then the protein is released to the cavity. These proteins either go into plasma membrane, secretory vesicles, lysosomes, or endosomes. In this way, the decision is made where to go the protein by the signal sequence. Now, what is the signal sequence? It's just like the pin code of a letter. Say every city is having a pin code. Suppose if a pin code is there, then that according to that pin code, the postman sorts the letters and then distributes. So similarly here, according to the signal, the proteins are distributed, sorted. Okay. This is normally proteins, they are chain of amino acids, but they will not be in the form of chain. They fold to form a different structures, which is called the primary structure, secondary structure, tertiary structure, and quaternary structure. For example, you see the alpha helix. Alpha helix is secondary structure. The myoglobin, that means alpha helix again folds itself on and forms a globular protein, that is myoglobin. That is called as tertiary structure. For example, in hemoglobin, you have 4 chains of globular proteins, 2 alpha and 2 beta. That is called as tertiary structure. So there will be different structures of protein, levels of organization of the protein. So here, this is the chain, unfolded protein chain, and it has 2 terminals, amino terminal and carboxy terminal. This is the folded protein. So on the amino terminal, you can see this red one. This is the signal for entering into the endoplasmic reticulum. The signal may be present at the terminal, or it may also be present in between as a patches. Now here you can see import into nucleus. That means if a protein that is synthesized in the cytoplasm has to go into the nucleus, then it should have the amino acid patches of amino acids having lysine, arginine, and lysine. So if you know about the amino acids, they are aliphatic as well as aromatic. And in aliphatic, you have leucine, isoleucine, valine. These amino acids, they are called as hydrophobic amino acids because they don't they dissolve in water. Whereas this, you have aromatic amino acids like say tyrosine, tryptophan, and phenylalanine. These are called as aromatic amino acids. So then you have acidic amino acids like glutamic acid and aspartic acid because they have two carboxyl groups. And then you have basic amino acids like lysine and arginine. So this is just classification of amino acids. You can refer afterwards. But here, for import into the nucleus, the signal is lysine, arginine, lysine. That means basic amino acids are required. Why basic amino acids? See, you might be knowing about histone proteins. Histones are present in nucleus along with the DNA. So these histones are basic proteins. So basic proteins means they will be having more of lysine and arginine. So the proteins which are having lysine and arginine, they are transported or imported into the nucleus. And export from nucleus, any protein that should come out of the nucleus means it should have hydrophobic amino acids like leucine, and isoleucine. So these are leucine and isoleucine, they are called as hydrophobic amino acids. And import into mitochondria. So if the protein has to be there in mitochondria, it should have to go into mitochondria from cytoplasm. So it should have this basic amino acids, but at a different sequence, not continuous sequence. It's in the nucleus, you have lysine, arginine, lysine. But here you can see arginine, lysine, but far apart. Important to plastids. For going in the plant cell from cytoplasm into the chloroplast, the protein should have serine, these amino acids. That is, serine contains like a hydroxyl group actually, it's water soluble. And important peroxisomes, for that you have hydrophobic amino acids at the carboxy terminal. The CO minus indicates carboxy terminal. Then import into endoplasmic reticulum. That is, just now we have discussed about the ER sequence, signal sequence. Import into endoplasmic reticulum. At the amino terminal, you should have hydrophobic amino acids. You see here leucine, valine. So these are all hydrophobic amino acids. And return to ER from cytoplasm, you see, from going out and coming in. So again, you should have acidic amino acid, aspartic acid, so glutamic acid. In this way, the different signals, they indicate where the protein should go. This is called as sorting signal, and that is called as targeting of proteins. Now, how the signal is recognized? For recognition of the signal, you have a particle protein that is called a signal recognition particle. So here, this is the signal recognition particle which recognizes the signal sequence. The SRP means signal recognition particle comprises of 300 nucleotides of RNA and 6 proteins designed. So this is the structure of the signal recognition particle. So this signal recognition particle recognizes the signal and then directs the ribosome to the endoplasmic reticulum. Now here you see how the synthesis of secretory proteins and their co-translational translocation across the endoplasmic reticulum. So this is the membrane, lipid membrane, and this is the cavity of endoplasmic reticulum. Now this is the ribosome. So when the ribosome starts synthesizing the protein, at the end terminus, if it has a signal sequence for endoplasmic reticulum, there is a sequence of hydrophobic amino acids, then this signal sequence is recognized by signal recognition particle. This signal recognition particle now then binds to the signal. Here you can see It is binding to the signal. And then it directs the ribosome to a receptor that is called as signal recognition particle receptor, which is present on the surface of endoplasmic reticulum. So because of this signal sequence and signal recognition particle, it binds to the receptor here. Here, this blue colored one is the translocon. That means it forms a hole for the translocation of the protein into the cavity of endoplasmic reticulum. Normally, it is closed. But when the signal recognition particle along with the ribosome comes and binds to the receptor, it opens. And you can see here, the new protein is continuously synthesized. This is translocon, closed when the ribosome complex is not bound to it, to the receptor. When the ribosome complex is bound to the receptor, it opens and allows this protein to be translocated through the pore into the cavity of endoplasmic reticulum. Now, at the adjacent to this translocon, you have an enzyme that is called signal peptidase. The signal peptidase cleaves or cuts off the signal sequence, and then the protein synthesis continues. You see here, and the protein comes into the cavity of endoplasmic reticulum, and this protein is folded into tertiary structure and released into the cavity of endoplasmic reticulum. So this is how the secretory proteins are synthesized in the cytoplasm, and they are translocated into the cavity of endoplasmic reticulum. Now coming to the membrane proteins. See, membranes, they have different types of proteins. On the surface of the membrane, you have proteins which are responsible for the transport of materials, which are called as transporters. Then you have receptors for enzymes, and then you have structural proteins. So in this way, you have different proteins, types of proteins in the membranes. So these membrane proteins, they're classified according to their arrangement. Now you see here, this is a protein with carboxy terminal, and this is amino terminal. And here you see, here you have alpha helix. So this protein is called as type 1 protein or glycophorin or LDL receptor. Say example is LDL receptor or glycophorin. Say LDL receptor is very much important. You know, LDL is called as bad cholesterol. HDL is called as good cholesterol. So if you don't have this LDL receptor because of genetic defect, then the LDL increases in the blood and leads to the formation of plaques and cardiovascular diseases like atherosclerosis. So all this is called as type 1 protein. In the type 1 protein, carboxy terminal is outside and amino terminal is inside. And this is a type 2 protein. So in type protein is quite opposite to type 1 protein. So here you have amino terminal outside, carboxy terminal inside. And these are the examples, transferrin receptor or glycoprotein receptors. All these things, they are example. So type 3 protein, you see here, the carboxy terminal is more inside here, whereas the carboxy terminal will be outside, but more outside. This is called as type 3. For example, cytochrome P450. This is present in the cytochrome P450 is important in endoplasmic reticulum for detoxification. Now here you can see the membrane protein is passing the membrane 7 times. So 1, 2, 3, 4, 5, 6, 7 times. So they are called as serpentine because of just like a snake. So the G protein coupled receptors or beta adrenergic receptors are of this type, or glucose transporter 1, which transports glucose from cytoplasm into the cells, or voltage gated calcium channels. All these are this type of proteins. So types of membrane proteins depending on the arrangement of the helix. Now, how these membrane proteins are synthesized and inserted. So membrane proteins are inserted when they have signal sequence. It just binds to the endoplasmic reticulum. The signal peptidase cuts off this cleavage signal, and then the protein, see, it is not released completely into the cavity. Instead of that, you see, here, this is the signal for stop transfer. This is transferring signal. Here, this is stop transferring signal. When the stop translated signal comes into the protein, these are also amino acids only. These are also amino acids. So then it stops the translocation, and you see here the protein is inserted into the membrane. And then it forms a vesicle, and this vesicle is transported to the membrane, which fuses with the membrane, and it is incorporated into the membrane. This is the incorporation of type 1 protein into the membranes of the cell. Now, type 2 protein. See, if you see type 1 protein, what is happening here? The carboxyl terminal is outside, whereas the amino terminal is inside. In type 2 protein, it is quite opposite to that. The amino terminal is outside and carboxyl terminal is inside. Now, this is the translocon. This is the signal for anchoring sequence. Then when this stop signal comes, it stops producing into the cavity, and then it releases into the membrane. Now, you see amino terminal, carboxyl terminal. Now, this also incorporated into the membrane. This type 2 protein. In this way, you have different signals. First, type 1 signal sequence, and in the middle, you have stop signal. Now, type 2, you don't have signal sequence, but you have a stop signal. Now, last one, just now I discussed about this beta adenine receptor. It has 7 stop and start signals. 1, 2, 3, 4, 5, 6, 7. So that it comes and goes in the membrane 7 times. Yeah, this is the example of that. So in this way, the membrane proteins are inserted in the endoplasmic reticulum. Then from the endoplasmic reticulum, a small vesicle is pinched off, which travels into the surface of the cell membrane and fuses, and this membrane protein is incorporated to the cell surface to the receptor. So receptors and all these things, they are incorporated like that. Now, in the endoplasmic reticulum, the protein is modified. Now, you may be knowing about the blood groups. Now, what are the blood groups? They will be having carbohydrate moieties, carbohydrates extra. To the proteins or lipids, carbohydrates are attached. Because of that, you will get the blood groups. So here, this blue one is N-acetyl galactosamine. This green one is N-acetyl glucosamine. This red one is fucose. And the brown one is galactose. So because of these things, you will get the blood groups. For example, in O, here, you don't have any extra. That's why it is called as 0, O or 0, you can say. In B, you have an extra galactose here. In AB, you have, in A, you have an extra N-acetyl galactosamine. In AB, you have both these two. That is, you have galactose extra and also N-acetyl galactosamine. That's why it is called as AB. So this is, if you don't have any, it is O. If you have only galactose, it is B. If you have only the N-acetyl galactosamine, it is A. And if you have both, it is called as AB. So this is how the blood groups are decided. And these carbohydrates are attached in the endoplasmic reticulum. So here you can see this is the amino acid chain, this protein chain. In that you have an amino acid called asparagine. This asparagine contains an extra amino group. So this extra amino group, these carbohydrates are attached. So this is galactosamine, N-acetyl galactosamine, mannose. So this is the chain. So you can see this is how the oligosaccharides, they are called as oligosaccharides because few chains, not long chain, small chains of carbohydrates are attached to the nascent proteins, that is newly synthesized proteins in the cavity of rough endoplasmic reticulum. This is called as protein processing. So this is all how, this is the mechanism how these different proteins are attached. There is a dolicol phosphate, this is a lipid, or to the protein chain. You see this is asparagine, to that the N-acetyl glucosamine, then mannose, then glucose. They are continuously attached by different enzymes. These are all enzymatic mechanisms. This is called as the processing of the proteins inside the cavity of endoplasmic reticulum in human. Now, formation of disulfide bonds. For example, some proteins will be having bonds which are called as the disulfide bonds between, say here, the disulfide bonds you can observe in, say, insulin. So formation of disulfide bond, how? This is the protein having the amino acid cysteine. The cysteine contains thiol group. Thiol group is SH group. So this is an enzyme that is called as oxidase PDA. So here the thiol group binds to this protein. Then afterwards he binds here. Then these two, they form a bond and oxidized substrate protein. So here you can see the formation of disulfide bond. So if you see in the insulin alpha chain and beta chain, you can find such structures. Actually, insulin is a one single chain. Actually, you have two chains, alpha chain and beta chain, but when it is synthesized, it will be synthesized as a single chain. Then afterwards, a small portion is removed so that you'll be having two chains. So here, target organelle, for example, after synthesis of protein, if it is endoplasmic reticulum lumen, the location of sequence, that is signal sequence, should be at the N-terminus, and the signal is removed. Removal of signal, yes. Then nature of sequence, core of 6 to 12 hydrophobic amino acids, often preceded by one or more basic amino acids. Suppose it has to go into mitochondria, it should have a signal sequence in the N-terminus. Signal is removed, yes. Then it should have 20 to 50 basic amino acids, arginine or lysine. Or chloroplast, location of the sequence, N-terminus. Removal of sequence, yes. There is no common motifs. It will be rich in serine. There is amino acid having hydroxyl group. Then into peroxisomes, the C-terminal, you should have signal. And then removal of signals, the signal is not removed. So it requires this serine, lysine, leucine at the extreme C-terminus. So in this way, so for these compartments, the protein should go into these compartments. They should have these sequences, amino acid sequences. Now, there are the examples of, for example, alcohol dehydrogenase 2. It is present in the matrix of mitochondria. Okay, so it will be having a matrix sequence, matrix targeting sequence. Then cytochrome oxidase present in the inner membrane of the mitochondria. So they have a matrix targeting as well as hydrophobic stop transfer sequence. ATP synthase. ATP synthase is present in mitochondria which synthesizes ATP. So it should have a internal sequence plus two internal sequences. ATP ADP antiporter. So ATP ADP antiporter means a protein which exchanges one ATP to one ADP. ADP is produced in cytoplasm after utilizing the ATP. It comes into the mitochondria and then ATP is transported one ADP to one ADP exchange. That is called as ATP ADP antiporter. Antiporter means it transports in opposite direction, one ATP to one ADP. Then cytochrome B2. This is also mitochondrial. No, this is a matrix targeting sequence at the amino terminus. Then in between, you have secondary cleavage by protease, the inner membrane space. So in this way, you have different signals for the same mitochondrial protein. Depending on that, either it should go to matrix or into the inner membrane or into the outer compartment, like that. In this way, so most mitochondrial proteins have a internal matrix targeting sequence. This is pink in color. That is similar but not identical in different proteins. So proteins destined for inner membrane, the membrane space or outer membrane have one or more additional targeting sequences. So for example, this ATP ADP antiporter is inner membrane protein. This alcohol is matrix protein. So the cytochrome C is intermembrane space protein, whereas porine is outer membrane protein. So you can see different signals for different proteins of the same mitochondria. So here you can see the amino acid chain. So how they are arranged. Now see how the protein is imported into the matrix of mitochondria. Now here you have the protein chain that is synthesized. Then you have a matrix targeting sequence of this pink color at the amino terminus. So this helps in binding to import receptor in the outer surface of the mitochondria, outer membrane of the mitochondria. Then it directs into translocon. From translocon, now you see these two translocons, they align in one direction, and then the protein is imported directly into the matrix of mitochondria. Matrix means inside the inner membrane. Here, this is cytoplasm. This is outer membrane of mitochondria. This is the inner membrane of mitochondria. And this is the intermembrane space. And this is a matrix. This is the mechanism how protein is imported into the matrix of mitochondria. Now, protein import matrix of mitochondria requires a site that is called the contact site. Here you see outer membrane and inner membrane, they are coming nearer and forming an attachment. This site is called as contact site. And through this contact site, the protein enters through the translocation into the matrix. This is the electron microscopic structure of mitochondria. You can see here. This one is upper membrane. This is outer membrane. This is the inner membrane. And this you see this site where the outer membrane and inner membrane meets, from where the protein is imported into the matrix. So this is this contact site. Now, how the protein is inner membrane protein. See, mitochondrial inner membrane protein, how it is assembled. This is outer membrane, inner membrane, but it has two sequences. One at the amino terminal, other one is a stop sequence. So when it starts entering into it, this signal is cleaved, and then when this stop sequence comes, automatically it is stopped. So that it remains in the membrane itself. So you can see here it is in the membrane. Targeting into intermembrane space. That is, this is the inner membrane, outer membrane. This is the membrane protein of inner membrane protein. Now this is the outer compartment is called, as you can see. This is matrix. This is the compartment, intermembrane space, where a cytochrome C is present. Now you see here matrix sequence, then intermembrane space targeting sequence. So because of that, it will not go into the matrix, but it remains inside between the inner membrane space. Important peroxisome. So it requires a receptor. For peroxisomes, it requires a receptor that is called as peroxisomal matrix protein receptors. This is the receptor. So this is signal sequence. It binds here, and then it exchanges the protein by a translocator. Now this is the secretory pathway. You can see here, this is the lumen of endoplasmic reticulum. These are the protein synthesis where the polysomes are there. Now they are attached to the ER membrane because of the signal. Then the signal is cut off and the proteins are released. And these proteins are formed into small vesicles. And these vesicles they travel from endoplasmic reticulum to Golgi. This is called as cis Golgi. From cis Golgi, here different modifications take place like attachment of carbohydrates and other things. Then you see it goes to trans Golgi. From trans Golgi, here the secretory vesicle is formed. And from this secretory vesicle, it is released to outside. The release is either regulated or continuous in the regulated secretion. So whenever, for example, in the case of insulin, when the glucose concentration is high, then only it is released. Similarly, when unregulated, it is released continuously. Now, then will be vesicles will be there, coated vesicles and even protein trafficking. So this is slightly complicated. The signal sequence, for example, lysine, aspartic acid, glutamic acid, and leucine. This is called KDEL signal. This KDEL signal is present. The protein with the signal, ER resident proteins. That means they reside only in endoplazmic reticulum. And if the lysine XX is there, ER resident membrane proteins. This is in the lumen. This is the membrane proteins. Diacidic, that means aspartic acid and glutamic acid is there. Then cargo membrane proteins in the cytoplasm of endoplasmic reticulum. If mannose-6-phosphate is there, attached to the protein, it goes into the lysosomes, lysosomal enzymes. Asparagine, proline, and tyrosine, for example, LDL receptor, it goes into the plasma membrane. Tyrosine, XX, and any other amino acid, membrane proteins in trans Golgi. Leucine and leucine, plasma membrane proteins. So in this way, different amino acid signals decide where should the protein go. And they should have receptors. For example, for this, you have a KDEL receptor. For this one, you have a COP1 receptor. For this one, you have a COP2 receptor. And for mannose-6-phosphate receptor for lysosomal enzymes. So it is very complicated actually. So you should have amino acid sequence as well as receptors for directing the protein into the proper sequence. So this is how the protein goes into the lysosomes. Now here, this is rough endoplasmic reticulum. The protein is synthesized and then it is coated with a COP2 coat. And when COP2 coat is there, it goes to ER, endoplasmic reticulum, to Golgi. In the Golgi, it is processed. And here, it binds to a receptor called KDEL receptor. From there, it comes into the lysosomes. So in this way, this is the electron microscopic structure of secretory proteins. So this is the plasma membrane. And this is the vesicle. And this vesicle, you can see, it is releasing the contents outside by fusing with the plasma membrane. This is a secretory protein. Now, the clathrin-coated vesicles are important because whenever the cells take, you know about pinocytosis and phagocytosis. For formation of a phagocytic vesicle, it should be coated with a protein that is called as clathrin, red one, this clathrin. When the clathrin coated is there, then this vesicle moves into the cell. And with this, the COP-coated vesicle, which contains the lysosomes, it fuses, forms late endosome. And here, the contents are digested. That is called as intracellular digestion. So this is how they target into lysosomes. So this is, and here, this is a lysosomal enzyme protein. And you see the signals here. With the signals, they bind to the receptor. That is the glucose N-acetyl phosphotransferase is the enzyme. This is the catalytic site, which adds phosphate group. And then, so this phosphate, because this phosphate group, it can bind to the KLDL receptor and enter into the lysosomes. Now, this is about the protein sorting. The protein sorting means these things are very complicated because you require, for this is the LDL receptor actually. So this is the LDL receptor. LDL receptor, you know, LDL is the low density lipoprotein. Actually, it carries the cholesterol from peripheral blood to the liver. And here we have, this is the LDL receptor. This yellow one is LDL, low density lipoprotein. And when the LDL binds to this LDL receptor, you know, a coated pit is formed. This red one is clathrin. Then it is invaginated. And after invagination, this meets with the lysosomes. And lysosomes, inside the lysosomes, so it is digested. Cholesterol, this is the cholesterol, and these are the amino acids. And then you can see recycling. This receptor is again recycled and comes into the surface. Again, it binds to the LDL receptors. So if the LDL receptors are absent in a person, then the LDL level increases in the blood, causing hypercholesterolemia, leading to the formation of cardiovascular diseases. So this is about the protein sorting, which I wanted to tell. So if you have any doubts about this targeting and all these things, I have skipped from some slides. So if you have any doubts, you can ask. Hello? Chat one. Okay. Any doubts?
Hello? 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 Hello, sir? Hello?
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 Hello? Hello, sir? Hello, sir? Hello?
Hello?
Hello? Hello, sir?
Yes, yes, నాకు వినిపిస్తుందమ్మా, మీకు వినిపిస్తలేదా?
వినిపిస్తుంది సార్. సార్ ఇది చిన్న డౌట్.
చెప్పండి.
How can we identify the terminating one and processing one, sir? మీరు చెప్పారు కదా, చూడడానికి రెండు పిక్చర్స్ ఒకలాగే ఉన్నాయి కదా, స్మాల్ హెడ్ పైన బిగ్ హెడ్ కింద.
ఎక్కడండి
మీరు ప్రోటీన్ ది అది చెప్పేటప్పుడు సార్, మనము అదేంటి ఎండ్ దాన్ని అంటే ప్రాసెసింగ్ స్టార్టింగ్ పార్ట్ ని
అమైనో టెర్మినల్ అంటారు దాన్ని,
సింథసైజ్ చేసే ప్రోటీన్ ని డిఫరెన్షియేట్ చేయొచ్చు ఇలా ఉంటే ఎండ్ అని చెప్పారు కదా, అదే ఇనిషియేటింగ్.
అవును,
అది ఎలా ఐడెంటిఫై అంటే చూడడానికి అయితే రెండు సేమే ఉన్నాయి కదా సార్.
మనం చూడగానే see when the amino acids are attached they form a peptide bond, and at one end you will be having amino group, that is called amino end.
Yes, sir.
To the other end you will be having carboxylic acid, that is called carboxylate.
Okay.
You see the protein structure has two ends.
Yes, sir. సో అమైనో ఎండ్ ఉంటే టర్మినేటింగ్, కార్బాక్సీ ఉంటేనేమో ప్రాసెసింగ్.
కాదన్నమాట, కాదు, అమైనో టెర్మినల్ లోపల సిగ్నల్ ఉంటే, ఫర్ ఎగ్జాంపుల్ a signal have continuous hydrophobic amino acids, then it is recognized by a particle that is called as signal recognition particle. This signal recognition particle directs the ribosome to bind to the endoplasmic reticulum at a receptor.
Okay, sir.
So for that you should know about biochemistry of proteins.
Okay, sir.
What is amino end, what is carboxylate end, how the peptide bond is formed, what is the structure of protein, what is alpha helix, all these things.
Okay, sir.
Sir, there are 6 charts in chat box, sir. Hello?
Yeah.
Thank you, sir.
Okay, welcome.
Please answer them.
Sir. Yeah. Chat box.
Let me go into that.
ఈ చాట్లు ఏంటి కనిపిస్తలేదండి.
There is no.
I'm not able to find anything in chat box.
Yes, sir. There is no questions.
Only that is allow to open speaker. Send PPT. Hello, sir. Send PPT. What is this? Only these things are there.
Yes, sir. Yes, sir.
Okay, sir.
You will share the PPT now, sir?
Yeah, they will be recording, isn't it?
Okay, sir. Okay, sir. Thank you.
You're welcome.
Okay, sir. Thank you very much for your extensive quality lecture. And on behalf of Department of Zoology,
because it's slightly complicated. Recent one, and they should know.
Yes, it is more complicated, and strength is also very bleak. Out of thousands of students, only 28 to 32 have joined.
Such deep subjects, it's difficult for them to understand. That's the problem.
But it is also there in syllabus, sir. It is included in syllabus. They need to understand that also.
Follow.
Sir, do we have the same topic again, sir? Means it is little complicated from basic. Can we have once? Because
let us see. It is.
What is that? I don't know.
No, sir. Just now you have said that we have to understand that carboxylic and what is
First, you should the fundamental thing is you should know about the structure of protein. What is carboxy terminal? What is amino terminal? How peptide bond is formed, and how different levels of organization will be there. In proteins, you have different types, say alpha helix, then beta peptide sheet. Then you have tertiary structure, that is folding of protein.
Yes, sir.
Example, myoglobin. Do you know what is myoglobin?
I have heard, but I'm not remembering.
Okay, myoglobin is present in the muscle. It stores oxygen.
Yes, sir. Yes, sir.
Okay. Do you know about hemoglobin?
Yeah, blood, no, sir. Which will carry oxygen.
Not in blood, it is in erythrocytes. Okay. But how many polypeptide chains are there in hemoglobin? Do you know?
I think 5.
No, 4. 2 alpha and 2 beta.
Yes, sir.
Yeah, that's why I'm telling you, you should have fundamental knowledge.
Yes, yes,
the protein, actually, first it will be having a straight chain. When the protein synthesized, it will be a straight chain. But it forms a helical structure, just like a spring. That is called as alpha helix. This alpha helix again folds on itself to form a round structure that is called as globular protein. Myoglobin. Why it is called as myoglobin? Because it is a globular proteins. Okay. So, but myoglobin is a monomer. It contains only one chain. Whereas hemoglobin is a tetramer. That means it contains 4. And they have 2 alpha and 2 beta. And they interact to form a structure that is called as tertiary structure.
Okay, sir. Okay, sir. Thank you.
Welcome.
And dear students, same topic is available in our C2, course 2, Cell and Molecular Biology.
Yes, sir.
Once you go through that, you can learn more detail.
Okay, sir. Okay, sir. Thank you.
If you don't have any doubts, I thank the audience for their patient hearing and also thank the organizers of the B.R. Ambedkar Open University Zoology Department, particularly Dr. Ponna Srinivas. Thank you very much.
Thank you, sir. Thank you, sir.
I will now hand over the session to Ponna Srinivas, sir. For thanks.
I wish all the students the next seminar, because the subject is very important. Have chosen speakers based on their expertise. So you need to understand the situation and also get the benefit of lectures given by experts. And Professor Kumar sir, thank you very much from the Department of Zoology, Ambedkar University, and Dean Madam for giving chance to conduct webinars. And thank you very much, sir.
Thank you.